Hi and welcome back to Ease Genetics. In this video, we will discuss the principles of Mendelian monohybrid inheritance, Mendel's first law of segregation, and Punnett square. Gregor Mendel wasn't just any monk. He was a genius observer, a thinker, way ahead of his time. In an era where the mechanism of inheritance were a mystery, Mendel answered a bold question: how are traits passed from one generation to the next? His experiments laid the foundation for what we now call genetics, presenting the principles of dominance, segregation, and independent assortment. This is why he is called the father of genetics. So let's see what he did. Using pea plants, Mendel conducted experiments that revealed the basic laws of heredity. He observed patterns in traits leading to great discoveries. So what was his experiment? He chose the pea plant for his experiments and studied 7 traits: seed color, yellow or green; seed shape, round or wrinkled; pod color, green or yellow; pod shape, inflated or constricted; stem height, tall or short; flower color, purple or white; and flower position, terminal or axial. So as you see, all the traits he considered had only 2 phenotypes. He started by crossing true breeding plants with different phenotypes. Pea plants are self-pollinated plants. A self-pollinated or self-fertilizing plant is a plant that can transfer pollen from the anther, the male part of a flower, to the stigma, the female part of the same flower or another flower on the same plant. The result of this will produce homozygous offspring for the traits. So Mendel used 2 plants, one with round-seeded and the other with wrinkle-seeded plants, both homozygous as now we know. But remember, he had no idea, and this makes his work amazing. After he crossed the 2 plants, he saw all the offspring produced round seeds, showing the dominant phenotype. Then he left the offspring free to produce the second generation, F2. So the offspring were self-pollinated. In F2, he saw a ratio of 3 round seeds to 1 wrinkle seed. He got the same results for all the other traits he was studying. With a review, this is what it looked like. He chose 2 true-bred plants and parents shown in P generation, one with round and the other with wrinkle seeds, and crossed them. The first generation plants, F1, all had round seeds. Then he allowed the F1 plants to fertilize freely, which in P plants they fertilize themselves. This type of cross is shown with a cross in a circle, and in the next generation, F2, he saw for each 3 round seed there was 1 wrinkle seed. He did the same for the other 6 traits and got the same 3-to-1 ratio results in F2. Here we see the numbers he counted for each trait in the F2 generation and reported in his paper published in Nature journal, which is accessible on the internet. For example, for seed color, he counted 6,022 yellow seeds and 2,001 green seeds in F2 generation, which is very close to the 3-to-1 ratio. If you divide 2001 by 2001, you get the 1 ratio, and if you divide 6022 by 2001, it will be 3.009. So close to the 3-to-1 ratio. You can do the same for the others. They are all very close to the 3-to-1 ratio, and those with a higher number of total count, like seed color, a total of 8,023, and seed shape, 7,324 in total, are the closest to the 3-to-1 ratio. This is because the more offspring you have, the more accurate your results will be and closer to the expected ratio. So remember, the total number of offspring affects the accuracy of the results and analysis. Mendel's results are all very close to the 3-to-1 ratio, and the differences are not significant. With these results, he established his first law in genetics, the law of segregation, which says during gamete formation, the 2 alleles for a gene separate. So each gamete carries only 1 allele. So if a parent genotype is heterozygous, here shown with big R, small r, its gametes would carry either big R or small r. Now consider he did not know anything about genes, chromosomes, and DNA. This makes it amazing. What he did from 1856 to 1863 and published in 1866 was 36 years ahead of his time, and it was not until 1902 that his discovery was rediscovered by scientists. The Punnet square was developed by Reynold C. Punnet, a British geneticist in the early 20th century, as a tool to predict the possible genetic outcomes of a cross between two organisms. He built on Gregor Mendel's foundational discoveries about inheritance, illustration to demonstrate that traits are inherited through discrete units now known as genes passed from parents to offsprings. So what he did was, for a trait considering one gene with two alleles, he drew a 3-in-3 table. In the first row and first column, he wrote each parent's gamete showing the alleles they carry. Then he summed up one by one the gametes of the two parents and wrote it in the table. Here, considering the red and white color of a plant flower, crossing two heterozygous plants, big R, small r, you see the genotype and also the phenotypes of the offspring are mentioned in the table. Well, this kind of table is a little fancy. You can draw it a little simpler by drawing lines like an XO game, but instead of playing X and O's, write the alleles of both parents, one in the first row and the other in the first column, and it doesn't matter which parent you mention in the row and which in the column. And in the table, by mixing the parent gametes, as shown with flashes in 4 different colors, blue with blue, red with red, pink with pink, and green with green, you will get the offspring genotypes in the table. Or it could be even more simple like this if the cross consists of a heterozygous and a homozygous parent, which needs only 3 rows and 2 columns due to the parents' genotypes. Or like this, it's up to you. Makes no difference how you want to draw the table and where to mention each parent. So maybe you don't want to draw a table and just want to write the cross between the parents and mention the offspring below it. Under each parent, write its gametes and the allele it carries. Again, by using flashes, you can write the offspring genotype, but looks like this: red with red, blue with blue, black with black, and finally green with green. It's just like mixing each gamete of one parent with the gametes of the other parent, one by one. You can write the F1 offspring genotypes and under each mention its phenotype. If needed, sum those that are the same. A cross between a heterozygous and homozygous could be done the same. As you see, the ratio is 2:2, which equals to 1:1 ratio, and we can simplify it. But in some problems, it's better to consider the cross results like this. This means considering the results out of a total of 4. This is very important, which I will get to it in a problem-solving video. Now, this is the simplest way and get rid of the flashes, which makes it a little messy. Write the cross and write the gametes each parent produces under it. Here I gave a number for each gamete for each parent in red and blue. The left parent with red 1 and 2 and the right parent with blue 1 and 2. Now with a fashion order, start with red 1 with blue 1, then red 1 with blue 2, next red 2 with blue 1, and finally red 2 with blue 2, and you are done. If you choose to consider the gametes of each parent with first and second, it would be first with first, first with second, second with first, and second with second. That easy, and you're done. If you found it helpful, please give it a thumbs up. It really supports the channel and helps others discover this content. If you want to see more videos like this and keep learning about genetics, don't forget to hit the subscribe button and turn on the notification bell. Feel free to share your thoughts, questions, or suggestions in the comments below. Thanks again for your support, and I'll see you in the next video. Take care.